Hello, we are continuing to highlight the virtual research poster competition. Joining us today is Karl Schuchard. Hey, Karl, how are you?
Hey, I'm great, Kyle. How are you?
Doing well. So tell us a little bit about yourself.
Well, I am Karl Schuchard, as Kyle just mentioned. I just finished my third year of my PhD in Industrial and Systems Engineering over at the College of Engineering. I'm under the advisement of Dr. Rohan Sherwaker, and our group is the 3D Tissue Manufacturing Group. And within this group, we use computational methods, experimental characterizations, and then statistics to model and characterize new biofabrication processes that we have invented.
Interesting. So tell us a little bit about your poster.
So my poster really comprises a process structure characterization of a new 3D biofabrication process that we're calling 3D melt blowing for tissue engineering scaffolds. And in this process, we've synergistically united a non-woven textile manufacturing process with additive manufacturing, or fundamentals of 3D printing, if you will, to fabricate some novel fibrous structures, which we think will have some broad applications in musculoskeletal tissue engineering. There are various process parameters that are now novel thanks to this new collection system for this traditional manufacturing approach. And that's really what my poster is characterizing, are these new process parameters that this process of collection has engendered. So in the first aim of the poster, we take a computational approach to modeling the relationships between this fiber traveling through the air and its interface with a high-speed rotating collector. The second aim of the poster is experimentally characterizing this process and structure relationship by actually manufacturing the fibrous mats or larger scaffolds using a biopolymer, and then using scanning electron microscopy as well as a variety of image analysis methods to then quantify some of these critical quality attributes from fiber alignment and fiber diameter. And what we really find is that the experimental results mimic or really reflect the computational results that we observed, in that those two parameters, the deposition location, kind of back and forth, as well as the surface speed, have effects on the fiber diameter and then the fiber alignment. And then in the third aim, we sought to experimentally characterize the mechanical properties of these scaffolds. And there we're using a standard test method for uniaxial tensile testing, and we found, I guess the primary result there is really that the mechanical properties are also reflected in the fiber morphological properties, where scaffolds or samples that had fibers that were aligned along the direction of uniaxial testing proved to be stiffer and stronger, which makes a lot of sense. If you line those fibers up basically side by side and then pull them, they're going to be stiffer and stronger than if you had randomly oriented fibers. And then I guess in the future, our goals are really in further characterizing this collection process using the computational method. So then the final future direction that we'd like to pursue is characterizing some of the cellular responses to these different fiber morphologies, and especially in long-term cell culture, where we're going to observe how these cells are proliferating and then secreting extracellular matrix, and then what the biomechanical implications of that are.
Thank you for sharing. And if people are interested, they could find more information on the poster on that website. Thank you, Karl. Take care.
You too.